My name is Bob Nagele, professor of geriatrics at the New Jersey Institute for Successful Aging. I was originally trained as a cell biologist, but I've sort of evolved myself into a cell pathologist, and as a result have headed toward working on diseases. I started working on Alzheimer's disease back around the year 2000 and have been stuck there ever since. So what I wanted to tell you about is a little bit of information about some of the mechanisms on how the disease works. Obviously, it is a irreversible, progressive brain disease. It slowly destroys our memory and thinking skills. The risk of getting it, of course, increases with age. It is an old person's disease. In most cases, symptoms appear after the age of 60. There is such a thing as a familial Alzheimer's disease, which appears very early, sometimes in the 40s. This only represents about 4% of total cases of Alzheimer's. These cases are inherited, of course, and almost all of them are mutations in the presenilin gene. The Alzheimer's disease is not a part of normal aging. It is a fatal brain disease. In terms of the statistics, Alzheimer's is the most common cause of dementia in people who are 65 and older. There's about 5 million people walking around with Alzheimer's in the U.S. now. And for every 5-year jump in age, the percentage of people with Alzheimer's doubles. So if things continue the way they've been going, by the year 2050, it's expected that we'll have 13.2 million older Americans with Alzheimer's disease, and I have 2 red-colored ifs. And they are big ifs. So if the current numbers hold, which we hope it doesn't, and if no preventative treatments become available, which we hope that there will be, and that's the way we're going to be able to change that number. The prevalence is sort of scary. If you look at this bar graph, there are 3 bars. It shows that in people who are between 65 and 74, about 4% of those people have Alzheimer's disease. Between 75 and 84, it jumps to 19%. And at 85, it jumps to nearly 50%. So 50% of all people 85 and older have Alzheimer's disease, which is frightening. Alzheimer's disease is broken up into a number of stages, but in fact, the disease is a continuum. It seems to start, if you look at the left-hand side of the plot, it starts a decade or 2 before symptoms appear. So they don't even know that they have it, and it's cooking in their brain. Eventually, you get to the point where you start to show a little bit of amnesia, some cognitive issues, maybe some memory problems, and the brain tries to compensate for that, and usually does, and people sort of get used to it, so they're not so sensitive in being able to pick it up in their loved ones. But eventually, it's followed by a rapid cognitive decline after they've been diagnosed with Alzheimer's, and then eventually they lose their independent function. And so this is the normal course of the disease. The symptoms include confusion. There's disturbances in short-term memory, problems with attention, spatial orientation. Changes in personality are really apparent, language difficulties, and of course unexplained mood swings. These are the main symptoms. And among dementias in this country, Alzheimer's disease represents about 70%. Vascular dementia makes up somewhere between 15 and 20, and Lewy body disease or Lewy body dementia between 10 and 15%. There are risk factors that have been recognized, and there are some protective factors. The risk factors, of course, include age as number 1. ApoE4 genotype is one of them. Female, although this is more historical, I think this may change in time, but right now females have a higher incidence of Alzheimer's. Hypertension and elevated cholesterol and head trauma are vascular factors, and as we'll see throughout this talk, we're going to emphasize the importance of the vasculature and the decline in the integrity of the vasculature in causing the disease and allowing it to propagate. The protective factors include high education, of course exercise, and that's rooted in cardiovascular, and mental activity, which means that it is brain vascular. So if you use your brain a lot, you flood your brain with blood, and you have very good blood vessels in the brain. The pathophysiology of Alzheimer's is considered to be a synaptic loss disease. And it's not just all synapses, it's primarily the synapses that are cholinergic. And so it is a cholinergic deficit disease. You end up having a progressive loss, a selective loss of cholinergic neurons. You get a progressive decrease in acetylcholine, and you also get, and then that leads to an impairment of activities of daily living, behavior, and memory, cognition. because of the primary role that the cholinergic system plays in this, there are a number of drugs that have been used for treatment of Alzheimer's disease, and you can see a list of 5 of them here. The first 4 are drugs that work on the cholinergic system, blocking the acetylcholinesterase enzyme, and the last one is memantine, which is an NMDA receptor antagonist, and that's used for moderate to severe Alzheimer's disease. The thing about these therapies is that they just treat the symptoms. And so I liken them to caffeine. You know, you wake up in the morning and you're tired, sleepy, or yawning, and your brain feels a little mottled. And then what you do is you get a nice jolt with a cup of coffee, and then all of a sudden you're feeling better and your performance has improved. So these are performance enhancers, and caffeine does that. However, the 4 drugs that are anticholinergics do that. They make you feel better, they make you perform better, but it's only a temporary thing. They do not treat the disease progression. They are not disease-modifying drugs. All they do is they treat the symptoms just like caffeine treats your tiredness. So the problem has been is that there are no therapies that are available for treating the pathology or the pathological progression of the disease. And that really is the aim of all the future Alzheimer's therapies. They want to treat the pathology directly. And one of the problems is you got to first understand what the pathology is and how the pathology evolves before you're going to be in a position to be able to, forecast what drugs are going to be useful and actually to be able to design tests that will actually work. And so we'll spend a little time talking about the pathology. And the first slide is, let's go with the gross level. We take a look at the brain itself. So you see 2 images up here on the screen. The one on the left is a normal brain, and you can see all the gray matter jammed in. All the gyri are smashed together, trying to get as much gray matter in there as you can per unit area. And then you see the brain on the right, and that's an Alzheimer's brain. And you'll notice the big difference is that there's now space in between the gyral folds. And the space is due to the fact that neurons are dying. When the neurons die, they don't take up space anymore, so the gyral folds will shrink. And then you get these big grooves, and it's easy to identify these brains in images, in brain imaging. If you look at a cutaway view, here's a cutaway view. You can see on the left is a normal brain. On the right is an Alzheimer's brain. And on the left, you can see the ventricle, and the ventricle is, one of the things you'll notice on the right-hand side is that the brain is actually shrinking. It's smaller, and so it shrinks away from the cranial vault, giving rise to a larger fluid space between the cranium and the brain tissue. The gyri, of course, are pulling apart from each other. You can see them radiating out. And also, if you look at the boxes, there's a language box and there's a memory box. And in the normal brain, the language box is filled with stuff. It's filled with neurons, gray matter and white matter. Same for the memory box. But when you go over into the Alzheimer's side, you can see that these boxes are now filled with largely space. And that's because the neurons that previously did those things are dying and have died. So the question really becomes in Alzheimer's disease is that what is it that makes these neurons die? And so that's at the gross level. When you take a look at the microscopic level, there are some pathological hallmarks that are very well known, been known for a long time. And one of them is amyloid plaques, and the other is a neurofibrillary tangle. In the upper left-hand corner of this slide, it shows a single amyloid plaque, which is spherical, has a dense core in the middle, and we'll describe why that is. And it's a little larger than the size of a neuron, a single neuron. And there are many millions of amyloid plaques that you would find in an Alzheimer's brain. And on plate number C, which is right next to that, to the right, that shows the morphology of a neurofibrillary tangle. And if you notice the shape is actually the exact shape of a pyramidal neuron. And it's stained with antibodies against neurofibrillary tangles. But if you look at the lower right-hand image, which shows a neurofibrillary tangle, you can see it in white, you can see that the cell is filled with this tangle of hair. And the hair is actually hyperphosphorylated tau. And it's a cytoskeleton. So basically the cell becomes filled with a skeletal material, and it so over-dominates the cell that the cell dies, leaving the skeleton behind. Just like in a graveyard, if you dig it up, you just got bones left, and that's all that's there. Although when you look at them in histological section, it looks like the neuron's still alive, but it is not. It's long since dead. So these are the 2 hallmarks, amyloid plaques, neurofibrillary tangles. The plaques appear early in the course of the disease. Tangles appear a little bit later. And that's why the focus of a lot of the therapies has been predominantly turned toward blocking amyloid from depositing in the brain. So the real question for a research guy like me is, what is it? What is it that's killing our brain cells? Why do these cells die? And if you take a look at this sort of it's a drawing or a cartoon, you can see that on the lower right-hand side, there's a little box that says healthy cells. And shown in that box are several pyramidal neurons. You can see the shapes are pyramids, and this is the most predominant neuron that is in the brain, and also the predominant neuron that's affected by Alzheimer's disease. If you take a look at the cells carefully, you'll see that they have lateral dendrites coming off on the side. These are the lateral dendrites. And then there's a main apical dendrite, and these support a giant synaptic tree, so that the pyramidal neuron may have as many as 10,000 individual synapses. But you'll notice in this picture how smooth the processes are, how straight they are. Now, what happens in Alzheimer's disease is we take a look at the upper left. And here's that same neuron now in an environment caused by Alzheimer's disease. And what you'll notice right away is instead of having a smooth surface and having smooth processes, all the processes are, the cell looks like it's shrunken, like a withered old tree. And the processes are corkscrewed, which means they actually look like wine corkscrews. And the reason why that is because those processes, those corkscrew dendrites, support a large dendrite tree which has collapsed. And as soon as the dendrites die and collapse, the process will retract. And when it retracts, it naturally assumes a corkscrew shape because neurofilaments travel through them in corkscrews, so it turns it into a slinky. So when you're looking at the pathology or section of a brain and you're wondering whether the person has Alzheimer's disease, you look at the pyramidal neurons and you look for corkscrew dendrites, and that's sort of a telltale. Now, one of the reasons why this neuron is not doing so well is because it's sitting next to this big, giant orange ball, which is called an amyloid plaque. And that amyloid plaque contains a material known as A-beta 42. It is a peptide. It's 42 amino acids long. And I have a slide here that illustrates where it comes from. A-beta 42 is produced by probably about 20 different types of tissues in the body. The most conspicuous or most important one is skeletal muscle. And as you know, skeletal muscle occupies a substantial volume of your body. So if it's producing amyloid, then obviously there's a lot of it and it's dumping it into the blood. And so where the amyloid comes from is, you can see on the left-hand side, near the letter A in the upper left, this cylinder is taken to represent the amyloid precursor protein, or APP. That protein is 671 amino acids long, and somewhere along its length toward the bottom, you'll notice this pink cylinder. That pink cylinder is 42 amino acids long, and that is A-beta 42, the troublesome peptide that actually gives rise to amyloid plaques. So in order to get from APP, the big protein of 671 amino acids, to A-beta 42, you have to undergo two successive cleavages by two enzymes. One is beta secretase, and one is gamma secretase. Beta secretase cleaves it here on top of the pink beer can, and then gamma secretase cleaves it below. And now frees the A-beta 42 to contribute to plaques and deposit in the brain. If you look down at the lower right, that is a very low magnification section of brain tissue showing hundreds of amyloid plaques in the section plane, and that's only two-dimensional. So if you go three-dimensional, it means that there are millions of amyloid plaques that are found in the brain, so much so that the brain actually becomes dominated by the presence of plaques, which destroys the neuronal environment. So if you ask the question that we want to know is, how is amyloid deposited in the brain in the first place? So I have three pictures here that I'm showing you, and the one on the left is a picture of three neurons, and they're in the brain and they're doing fine. It's a healthy brain. And then I show you an Alzheimer's brain in the middle. And according to one theory, the amyloid plaque sort of seeds out like a crystal in the space between the neurons. And the idea is that that crystal or plaque will grow, get bigger and bigger and bigger, and eventually encroach on the neurons and kill them, strangle their processes, and so on. And that's most of the amyloid therapies that have been developed in clinical trials are based on that mechanism. Unfortunately, it's wrong. So I've modified the diagram to show you on the right-hand side what seems to really happen. And you'll notice the difference is that this neuron here at the bottom, on the right-hand panel has a load of intracellular amyloid in it. So the cell is depositing amyloid inside the cell. The cell becomes pregnant with this amyloid, and the amyloid actually gets in the way of cellular processes, and eventually it will become toxic, kill the cell will die and pop. And then if you take a look at the other two neurons at the top half of the picture, you'll see that I have replaced them with amyloid plaques. And the meaning of all this is that amyloid first accumulates in the neurons intracellularly, and then after a long time it will explode or pop and die. And when it dies, it releases the entire cloud of amyloid into a spherical structure that we now call a plaque, which is nothing more than a gravestone or a grave marker indicating where the cell used to be. So to prove that I'm not lying to you, here's some electron micrographs, high-resolution light micrographs of pyramidal neurons showing huge deposits of A-beta 42 within them. Here on the left-hand side, you see a single neuron at the bottom, okay? And there has a big nucleus, and below that nucleus is a giant deposit of sequestered A-beta 42. It actually occupies almost 50% of the total cytoplasmic volume, and that cytoplasmic volume used to have synthetic machinery that supported the dendrite tree. If you take half of the synthetic machinery away, you can only support half the tree. And that's why the synapses are lost during the amyloid accumulation period, which can extend for years. Here again on the right-hand side, you see a similar view. So if we are correct and we say that, gee, well, it looks like the amyloid plaques are actually the remnants of exploded neurons, individual exploded neurons, then that means that when you take a section of a brain that has a lot of amyloid plaques, you should not be able to find any neurons. And in fact, there's not one pyramidal neuron in this field of view. On the other hand, if it turns out that there are few plaques, in this case only one in this slide, you can see now the large pyramidal neurons on the right-hand side, and you'll notice that each of them has a dark brown deposit of A-beta 42 peptide accumulated within the cell. Pretty soon, when we come back to it, maybe a month later or 2 months later, it'll end up, all those cells will die and pop, and they'll look like this, which is now just a field of amyloid plaques. Now, as I mentioned to you, Alzheimer's disease is a synaptic loss disease. And so I show you on the left-hand side a normal pyramidal neuron that has 10,000 synapses. That's the average for that cell. Take a look at where the cell body is. It's a tiny little thing at the bottom of the slide. There are 10,000 synapses here. During the amyloid accumulation time, the cell will begin to lose synapses. So it goes from 10,000 to, you can see, 4,000 in this case. So because there's far fewer branches that are able to be supported. By the way, the cell is still alive and the amyloid is still contained within the neuron, yet it has less than half of the synapses that it had before. So I'm sure you understand why you can have amyloid deposition in the neuron and you have a loss of functional performance, okay? And yet the neuron is still alive. And that's why they've had some difficulty with some of the floor-beta-peer imaging, where they've done imaging of amyloid load. And at this stage, since the cell is still alive, the amyloid is contained within the cell body. When they use the floor-beta-peer or AV45 imaging agents to detect amyloid, they come up empty. They come up blank, and they assume there is no amyloid, when in fact, the amyloid is actually hidden within the bodies of the neurons and is inaccessible. Okay, so as things continue on, you drop down to 1,500 synapses, and then eventually the cell dies and it pops, and you'll get an amyloid plaque. From our point of view, we're always looking for a therapy to block amyloid deposition. And how do you do that? And you can do that, but you first have to know what is the source. And as I mentioned to you, A-beta 42 is made by a number of cells, including skeletal muscle cells. And we have so many of those. And they dump it into the blood. So the blood is a major source. Now, in healthy people, we have a thing called a blood-brain barrier. And the purpose of that barrier is to prevent things from getting from the blood into the brain and disrupting homeostasis. The brain works really hard to make sure that the environment in which the neurons are operating is pristine and optimized for their function. Anything that deviates that, any leaks in the vasculature, for example, or leak through the blood-brain barrier will alter homeostasis and can influence in a negative way the functional performance of your brain. So it turns out that we did some work a number of years ago, back in the early 2000s, and we found that there was a major effect on the blood-brain barrier in the case of patients with Alzheimer's disease. So let's take a look at what the blood-brain barrier is first. Here's just a cutaway view of a blood vessel. And the inner lining of the blood vessel looks like it has cobblestones. Each one of those cobblestones is a vascular endothelial cell, and the little p, the letter P, is pointing to the junction or the seal that occurs between them. These are tight junctions, like spot welds, and the cells are completely bound together so that nothing that's in the vessel can leak across that impermeable barrier. That's the way the blood-brain barrier is supposed to work. In older people, as we'll see, the barrier becomes compromised by a number of things. So these are sections showing you, if you look at the title, it says, in all Alzheimer's brains that I've ever looked at, the blood-brain barrier is chronically defective, which means it's leaking all the time. So at any one point, you can immunostain a slice of tissue and find out what was leaking at the time of death. And so here you see on the left-hand side, this is A beta 42 painted red. You see the artery in the middle of the field of view, and the red cloud around the outside is A beta 42, which is leaking out of that vessel. If you go to the one on the top right, that's complement C1q leaking out of this vessel, which you see in cross-section. And then down below, you see a low magnification view of two vessels, and the brown cloud that you see surrounding them is immunoglobulin G, because obviously antibodies are also going to be able to leak out of the vessels and get into the brain. And that's not a good thing. The question is, what is it that makes the blood-brain barrier leak? Now, this looks like a very complicated diagram, but it isn't. It just lists the things that can make the barrier leak, most of which will be common sense to you. You can have infections. For example, urinary tract infections are well known to cause delirium in the elderly. And delirium, as we'll see, is in large part due to blood-brain barrier leaks. Of course, aging is a risk factor, diabetes for sure, high cholesterol, which changes the integrity of the vascular lining, any stroke or traumatic brain injury, high blood pressure, because if you have high blood pressure and you have holes in your blood-brain barrier, it's going to push the fluid out under higher pressure into the brain. And the real big one for the elderly is anesthesia. That's really a bad thing. In any case, all of these contribute to a disruption of the blood-brain barrier, which you see in the red box. And then if that disruption occurs, plasma will leak out into the brain tissue, and it'll throw the neurons off in their function. Now, if it turns out that that loss of homeostasis is short-term, like for example, during surgery, you have a surgical anesthesia, and it's acute, you may get delirium, because delirium is when all these things leak out into the brain tissue and the neurons fire willy-nilly with no particular pattern, and the brain behavior is almost unexplainable. We do recover from surgery, though. We're all wackos to a certain degree after surgery, so that's why when we wake up, we're a little groggy or not making any sense. But of course, the barrier then immediately shuts if you're healthy, and then you can be fine. But if it turns out you're an older person and you were teetering on the edge of a blood-brain barrier breach anyway, then this will continue, and the leak will actually promote the development of dementia caused by your surgery initially. Now, it turns out that the blood-brain barrier can leak anywhere. So actually the site at which the leak occurs is important. Of course, the extent is important. But let's say, for example, it happens in the substantia nigra area. Well, if that's where the leak happens, then you end up with Parkinson's disease. If the leak happens up in the frontotemporal region, you end up with frontotemporal dementia. If it ends up in the hippocampal entorhinal cortex, you end up with amnestic malocognitive impairment. So it all depends on the site or the location of the leak, which of course we can't control. Here's a study that we did on anesthesia, which is really a bad thing because of the post-surgical delirium that you get. Basically what it shows you is that when you take a look at the left-hand panel, that is a very, very high scanning electron microscopic view, many thousands of times in magnification, showing the surface of the blood vessel. Each one of those ridges that you see is it is actually the blood-brain barrier. You're looking at the actual tight junctions of the barrier. But notice what happens when you treat them with, or you have 3 hours of surgical anesthesia. What ends up happening is it looks like somebody got a rolling pin and rolled down all the hills. And also you'll notice that there are new things appear called holes. And these holes are breaches in the blood-brain barrier that leak. And if the vessel, of course, is an artery and it's under pressure, obviously it's going to be spraying fluid into the brain tissue, and you will have disruption of homeostasis. So there is the direct morphological evidence for what's happening as a result of anesthesia. And the worst offender is sevoflurane. Isoflurane's a little easier, and propofol is a little better. So when you try to convince people that the blood is the source of A beta 42 that ends up depositing in the brain, you got to be able to show it. And you can't use human brains to do that, so we're stuck with animals, so we use a mouse. And so you go to Walmart and you buy fluorescent green labeled A beta 42 peptide, and then you inject it into the tail of the mouse, where you can see the arrow touching the tail. And you also inject the mouse with pertussis toxin, which induces blood-brain barrier breakdown. Now, if the barrier is intact, you can inject all the fluorescent A beta you want, and it'll stay within the vessels, as expected. But if you break the blood-brain barrier down, then the green stuff will begin to leak into the brain tissue. So here you see on the left-hand panel, you see a section through the hippocampus of the mouse, and you see all this green stuff. On the upper left is where the leak is, but on the bottom or the lower right of that picture, you'll see individual green spots. These are individual pyramidal neurons that have affinity for A beta 42. So A beta 42 sticks to those neurons like flypaper and coats the surface of those neurons. And we'll see that that will be a problem later on. And so if you look at the picture here on the left again, here's a mouse brain pyramidal neuron. You can see the big dark thing in the middle is the nucleus. But see the little positive granules that we're pointing to at the base of the cell. That is A beta 42 that was internalized by those cells, and it's deposited in the same place that we see in the corresponding image on the right in the Alzheimer's brain from a human. So we think that this is a really good mimic for the human AD pathology model. One of the other things that leaks out that is really problematic, in fact, probably the major one, is shown here in this slide, is that if you take a look at the upper left-hand, the left-hand picture in the upper left-hand corner, there is a blood vessel. You can see that it's an open hole. From that vessel, there's a blood-brain barrier breach, and immunoglobulin is leaking out, forming a brown cloud that is moving to the lower right. And as it passes by those neurons, the immunoglobulin binds to the neurons and makes them dark. So you can see the dark neurons in the path of the cloud. And that means that the autoantibodies, these antibodies are binding to the neurons. And the question is, what is it that happens? How does the neuron respond to when an antibody is cluttering up its surface? And the answer is, whenever it binds immunoglobulin, it induces endocytosis and tries to pull the antibody inside. But remember I showed you that A beta 42 was also bound. So if the antibodies can induce endocytosis and drive the antibodies in, the A beta 42 takes a ride with it. And that's how the A beta 42 gets into the neuron progressively over time. Okay, and eventually the cell will accumulate it. Unfortunately, A beta 42, once it gets inside of a cell, cannot be degraded. So if you have something that's coming in all the time, and you can't degrade it, the end result is accumulation. And so you have no choice. So we think that it is brain reactive antibodies in the serum that are actually causing this. Up until now, these antibodies have not been able to get into the brain because we have a blood-brain barrier. I have a lot of them in my circulation that do react with brain neurons very well. The only reason why they're not doing it is because presumably I have an intact blood-brain barrier, hopefully. If I slur my speech, you know that's not true, right? So as we get older, if the barrier breaches, then the deal's off, right? Those antibodies can get in. So what we did here is we took mouse brain out, and you can actually slice the mouse brain up into slices like a loaf of bread, and you can keep them alive and functioning for 13 weeks. And so we wanted to ask the question: do antibodies push amyloid? Are they the reason why amyloid is depositing in neurons? And the answer is shown in this slide. So if you look at on the left-hand side, that is a section of cerebral cortex that was treated with 100 nanomolar, which is physiological, that's the concentration in our blood, of A beta 42 all by itself with no antibodies. And you can see that there's a little bit of a brown accumulation in a couple of the neurons, but nothing earth-shaking. But if you add human serum that has antibodies that will react with the neurons, you can see now how intense the brown color is. There's at least a 20-fold increase in the deposition of A beta 42 in those neurons. So again, we conclude that it's the combination of A beta 42 binding to the neuron and the antibodies pushing it in by endocytosis. So the mechanism is then summarized here. If you look on the left-hand side, you'll see this purple blood vessel. And the contents of the blood vessel are, obviously serum or plasma. And I have Ys to represent antibodies, and red dots are A beta 42. And the lower half of the vessel shows an intact blood-brain barrier. All the junctional seals are good, and everything is inside the vessel. But as soon as you disrupt the blood-brain barrier, as is shown in the top half of the vessel, you will end up having a leak cloud. And we saw some of those leak clouds in my histological pictures that I showed you earlier. But in that cloud will be A beta 42 red dots and the autoantibody Ys, which react to the surface of the neuron. Again, they go inside the neuron, the antibody's digested, but the A beta 42 accumulates over time. So that cell in the middle ends up moving to the cell in the right, which has all those red dots accumulating in its cell body, and it can no longer support the dendrite trees, so you have a dendrite collapse, loss of synapses, and then behavioral and memory changes. So that's the mechanism. And if that's the mechanism, then there is a cure. Where should you focus your attention developing a therapeutic? How about we stop the barrier from breaking in the first place, where the X is? Okay, if we block blood-brain barrier breakdown, then that means all those terrible events that happened on the right-hand side will not happen. Okay, and that is the goal for therapy, in my opinion.